Hello, my name is Minkyu Park, and I am working as a research associate in the Department of Horticultural Science at the University of Florida. First of all, thank you for inviting me to this seminar. And today's my talk is about sample sequence analysis uncovers recurrent horizontal transfers of transposable elements among grass species. So the goal of this study is the development of new method for comprehensive investigation of horizontal transfers. The horizontal transfer is considered as one of the important mechanisms for the evolution. However, its study on plants is not well established yet. The two major bottlenecks of the horizontal transfer study in plants are the limitation of available sequence resources and analysis methods. To solve these bottlenecks, I tried to develop new analysis method for horizontal transfer investigation using low-depth NGS sequences. In this study, I successfully developed the new method and could verify the method with previously uncharacterized plant species. The horizontal transfer is involved in the evolution of life. Evolution of life is an underlying phenomenon identified from all kinds of living organisms. In general, the evolution of life is known to occur through genome modifications such as genome duplications, substitutions, activity of transposable elements, and horizontal transfer of DNA. Therefore, understanding of the horizontal transfer is also important for understanding the evolution of life. This slide shows the definition of horizontal transfer. Depending on the DNA, direction of DNA flow, if the DNA is transferred from parents to progeny, we call it vertical transmission. This is the general mechanism of DNA transfer that we know very well. However, if the DNA is transferred from one species to another species across mating barrier, we call it horizontal transfer. Horizontal transfer phenomenon has been identified from diverse organisms such as bacteria, fungi, animals, plants, and even between different kingdoms. Whereas the study of horizontal transfer in bacteria is very well established, that in eukaryote is not well established yet. However, thanks to the increasing genome data, more and more horizontal transfer cases in eukaryotes are reported recently. The transferred DNAs by horizontal transfers in eukaryotes are like transposable elements, organelle genomes, and genes. The horizontal transfer phenomenon in plants is not well studied yet compared to other organisms. The overall horizontal transfer studies in plants are as follows. Horizontal transfer of organelle genome in plants is relatively well known in plants. It is known that the mitochondrial genome can be horizontally transferred by grafting, and this is one known mechanism of horizontal transfer in plants. In the case of transposable elements, it is relatively not well studied. According to the previous study, horizontal transfer of LTR-retrotransposons is widespread across diverse plant species, but the mechanism is not known yet. Therefore, it seems plants also use the horizontal transfer in their evolution, but because of the not active study, it is not well known yet. In the case of horizontal transfer of genes, only a few cases has been reported in plants, indicating it is a rare phenomenon. So in this study, I used two approaches to investigate the horizontal transfer in plants. One is LTR-retrotransposon-targeted investigations of horizontal transfers, and the other is non-targeted investigation of horizontal transfers. Because there was a previous study of plant horizontal transfer with LTR-retrotransposons, I developed the LTR-retrotransposon targeted investigation method based on the previous study. Through these, I could identify horizontal transfers of LTR-retrotransposons among grass species. Based on the success of the targeted method, I also tried non-targeted investigation of horizontal transfers. This non-targeted method was supposed to identify all kinds of horizontally transferred DNA, and I expected to find some horizontally transferred genes, but only transposable elements were identified as the horizontally transferred elements in this study. In this study, I used the low-depth sample sequences to identify horizontal transfers. The strength of low-depth sample sequencing in horizontal transfer investigation is that it enables the horizontal transfer analysis with low cost. Therefore, we can investigate the horizontal transfers in the previously uncharacterized genomes by generating low-depth NGS sequences. However, the disadvantage of this method is still it needs the fully assembled target genome sequences to identify the horizontal transfer. Also, because of this, the method is a little bit complicated to use. In contrast, the advantage of using reference genome sequencing in the horizontal transfer investigation is that it is easy to analyze the horizontal transfers. However, if we use the assembled genome sequences, the sequence materials for horizontal transfer investigation is highly limited, and the generation of new assembled sequence is very expensive. Therefore, these become the research bottleneck of horizontal transfer study. So this study focuses on solving the research bottlenecks of using the assembled whole genome sequences. Now let me talk about the development of LTR-retrotransposon targeted investigation method. This is the brief introduction of LTR-retrotransposon. The LTR-retrotransposon is the major transposable element that comprise plant genomes. Gypsy and Copiae are the two major superfamily of LTR-retrotransposons. If you see the right figures, the left panel shows the pepper packeting chromosomes, and the right panel shows the fluorescence in situ hybridization image with one of the gypsy elements of pepper. The green signals indicate the LTR-retrotransposons, and as you can see, it is the major component of the genome. And their structure shows the long terminal lipids at both ends, and the functional domains for their replication exist between the two LTR sequences. Among the functional domains, the reverse transcriptase is the most well-conserved domain, and I used this domain in the LTR-retrotransposon targeted investigation of horizontal transfers. So this slide shows the simplified version of LTR-retrotransposon targeted investigation. The first step is generating low-depth NGS sequences from the plant genomes for investigation that are expected to contain horizontally transferred LTR-retrotransposons. For LTR-retrotransposon targeted horizontal transfer investigation, the next step is mapping reverse transcriptase sequences in the sample sequences to the target genomes. The reverse transcriptase is the well-conserved functional domain of the LTR-retrotransposon. In this case, the target genomes are assembled genome sequences that are available from public genome database. With the 94% threshold of the mapped reverse transcriptase, the 40 kb sequence contigs containing the reverse transcriptases were isolated as the candidate contigs that may contain the horizontally transferred LTR-retrotransposon. After that, the whole sample sequence reads were mapped to the 40 kb sequence contigs. And based on the mapped density and identity, the purity of horizontally transferred LTR-retrotransposons were determined with the 97% identity threshold. This slide shows the detailed method for the LTR-retrotransposon targeted horizontal transfer investigation. In the first screening, the low-depth NGS sequencing was performed with 19 panicoid species by 3x coverages of the genome sizes. The sample sequences were then translated into 6-frame amino acid sequences. And using the hidden Markov model, reverse transcriptase containing sequence reads were isolated. And these reverse transcriptase sequences were mapped to a total of 115 angiosperm genome sequences. By 94 identity threshold of the mapped reverse transcriptase sequences, the 40 kb length of candidate contigs containing the possible horizontally transferred LTR-retrotransposons were isolated. In the second screening, the whole sample sequences were mapped to the candidate contigs. The mapped candidate contigs were filtered by 97% identity threshold and 5 reads per 2 kb of density threshold. The filtered contigs were filtered again if the mapped sequences contain conserved sequences like organelle genome, ribosomal sequences, simple repeat sequences, and highly conserved genes. After this filtering, the candidate horizontal transfer cases were confirmed by comparative sequence analysis and phylogenetic analysis. At the end, the horizontally transferred LTR-retrotransposons were classified and visualized for publication. To test the method, we generated sample sequences from 19 previously uncharacterized panicoid species. As shown in the picture, the panicoids are grass species, and some of them are known as weed plants of rice. This right figure shows the phylogenetic tree of the 19 panicoids, and the black bars shows the total size of the sample sequences. And these gray bars indicate the estimated genome sizes, and the average genome coverage of the sample sequences were about 3-fold. And this shows the result of LTR-retrotransposon targeted horizontal transfer investigation. I could identify a total of 4 horizontal transfer cases between the 4 panicoids and Oryza sativa. The species distance between the panicoid and Oryza were estimated by identity histogram of orthologous CDS, and the peak appeared at 86%. This graph shows the map sample sequences to the target genome contigs. The horizontal red bar indicate the target genome contigs, and the vertical bars indicate the map sample sequence reads, and this height is proportional to the identity. The horizontal dotted red line indicates the 97% identity threshold used in the horizontal transfer determination. Considering the 86% of the species distance, the 97% of identity threshold was high enough to identify the horizontal transfer. Because the horizontal transfer were identified only from Oryza species among the 150 angiosperm genomes, we further investigated horizontal transfer with 10 different Oryza species that have assembled whole genome sequences. Using the same method, we could identify a total of 15 horizontal transfer cases between 19 panicoid and 10 Oryza species. In the figure, phylogeneity of panicoid and Oryza species are shown, and the species that have horizontal transfer cases are connected by lines. To validate the horizontal transfer, we used phylogenetic analysis using the reverse transcriptase homologs. And the reverse transcriptase homologs of horizontally transferred LTR-retrotransposon was isolated from both donor and recipient species, and the phylogenetic tree was constructed with these homologs. If you see this figure, the filled red diamonds indicate the reverse transcriptase homologs from Oryza punctata. And the empty diamonds indicate that that of the Echinochlora pyramidalis and Echinochlora haploplata. If there is no horizontal transfer between the Echinochlora and Oryza, the reverse transcriptase homologs have to be clearly separated. However, 2 homologs of Oryza punctata were identified in the Echinochlora cluster. This indicates that one of the Echinochlora LTR-retrotransposon was horizontally transferred to the Oryza punctata genome and proliferated into 2 copies. This figure shows the validation of horizontal transfer by comparative sequence analysis. The horizontal red bar indicates the sequence contig of Oryza novara that contains the horizontally transferred LTR-retrotransposons. The horizontal yellow bars indicate the collinear sequence contigs of other Oryza species. These vertical bars indicate the mapped panicoid sequences to the Oryza novara sequence contig. As shown in the figure, the horizontally transferred LTR-transposon appears as a new insertion in the Oryza novara sequence, and the same sequence, the collinear sequence in other Oryza species, doesn't contain these newly inserted LTR-retrotransposons. So this clearly shows that this LTR-retrotransposon in the Oryza novara is horizontally transferred from the panicoid species. Among horizontal transfer cases, some showed the horizontal transfer to the common ancestor of the recipient, and this slide shows the case. An LTR-retrotransposon of single-pogon stratus was horizontally transferred to the common ancestor of Oryza sativa and Nevada. Although it was not detected in our horizontal transfer investigation, Oryza rufipogon also appears to contain the horizontally transferred LTR-retrotransposons. The phylogenetic tree validates the horizontal transfer of the LTR-retrotransposon to the common ancestor. The cluster of LTR-retrotransposons of the three Oryza species were nested in the cluster of the Symbopogon stratus cluster. Therefore, this also supports the true positive result of the horizontal transfer investigation. This table shows the summary of all the 12 horizontal transfer events identified in this study. Interestingly, 5 out of the 12 horizontal transfer events were identified between Echinocloa and Oryza species. This indicates that the horizontal transfer may occur actively between Echinocloa and Oryza species. Therefore, we investigated more horizontal transfer with these Echinocloa species. At this time, we tested a non-targeted horizontal transfer investigation method with Echinocloa and Oryza species. For this, we generated additional sample sequences from 4 additional Echinocloa species. This shows the overall method of the non-targeted horizontal transfer investigation method. Different from the targeted method, we met 100 megabase size subset of sample sequences in the first screening. All the rest methods are identical with those of the targeted method. This shows the phylogenetic tree of Echinocloa and Oryza species used in the non-targeted horizontal transfer investigation. Both Echinocloa and Oryza belong to Poaceae. The investigation of non-targeted horizontal transfer were performed between 9 previously uncharacterized Echinocloa species and 10 Oryza species that had assembled whole genome sequences. And this shows the representative horizontal transfer cases detected by non-targeted investigation. The horizontal red bars indicate the sequence config of Oryza genomes containing the horizontally transferred DNA. The vertical bars indicate the sequence. Vertical bars indicate the mapped Echinocloa sample sequences. The densely mapped region in the middle of the bars indicate the horizontally transferred DNA. The blue boxes in the red bars indicate the LTR retrotransposons, and the black bars are the DNA transposons. Although it was non-targeted horizontal transfer investigation, only transposable elements were identified as the horizontally transferred DNA in this study. And this shows the number of horizontal transfer cases by different thresholds. So these are the identity thresholds, and these are the number of horizontal transfer cases. And as you can see, the number of horizontal transfer cases is proportional to the identity threshold. This indicates that The horizontal transfer between the two genera is a recurrent phenomenon. And this table shows the classification of 165 horizontal transfer cases identified by the 94% identity threshold. LTR-retrotransposons were most abundant as a total of 109 cases, and the number of copia was 70 among them, which is the most abundant among the repeats. A total of 49 horizontal transfer cases of DNA transposons were also identified. Although horizontal transfer of line elements is known to be very rare, our investigation method identified 3 cases of horizontally transferred line elements. Because horizontal transfer cases include the parallels of the horizontally transferred elements, we clustered the 165 horizontal transfer cases to identify the horizontal transfer events. As a result, we could identify a minimum of 48 horizontal transfer events in our study. Interestingly, massive horizontal transfer events occurred between Oryza punctata and Echinoclea, and most of the horizontally transferred horizontally transferred DNA transposons were identified in Oryza punctata. And the activity history of the parallels in the clusters were calculated. From the result, we could know that the horizontally transferred transposable elements were actively proliferated after integration into the recipient genome. Because we used a high threshold to find the horizontal transfer cases, we also expected to find the direct donor and recipient species. To investigate the direct donor and recipient species, we compared the identity of the horizontally transferred transposons between the donor and recipient species. And this heat map shows the red color density is proportional to the identity of the horizontally transferred elements. And interestingly, we could find three cases of horizontal transfer that shows 100% identity between Echinoclea calopus and Oryza brachyantha. This figure shows the mapped Echinoclea calopus sample sequences to the Oryza brachyantha sequence config. As you can see, all the identity of the mapped sequences is 100%, indicating the two species are the direct donor and recipient. There were 3 parallels of the horizontally transferred elements, but the 2 of them were identified as the solo LTR, which are the remnants of the parallels of the horizontally transferred LTR-retrotransposons. The geographical distribution of the Echinoclea calopus and Oryza brachyantha was also same as the Middle Africa. As you can see here, they show exactly the same distribution area. So this also supports the 2 species are the direct donor and recipient of the horizontal transfer event. So this is the summary of the presentation. In this study, I could develop a new method to investigate the horizontal transfer and validate that the comprehensive investigation of horizontal transfer is possible with low depth sample sequences. Using the new method, we could identify that the recurrent horizontal transfer of the multiple transposon families occurred between Echinoclea and Oryza species. And this was expected to contribute to the transposable element diversity in the host genome. And Oryza brachyantha and Echinoclea calopus were determined to be the direct donor and recipient species of horizontal transfers. Therefore, these species could be used as model species to investigate the mechanisms of plant horizontal transfer. So thank you so much. And if you have any questions, please send it to me through this email address. Thank you so much.